Okay, so I wanted to start today with the with some of the different models. So we had talked about basically at the beginning of the semester or towards the beginning of SI this semester, we did most of the models. And so I want to finish those up today before we move on and talk about meiosis. Okay, so are there any questions at all, people who basically were working on the tissues packet over the weekend? Any questions on that at all? Not actually sure. Oh, we're on Thursday. Sorry, I don't even know what day it is. So remember, as always, if you guys have questions, remember to write me in Canvas. And then also, obviously, we still have the rest of our SI session today. I'm not able to do tutoring today. So usually every week I do tutoring from 11 to 12 right after this session. But this week I have a very busy schedule. And so unfortunately, I'm not able to do tutoring today. So if you have any questions, please write me a Canvas inbox. And, you know, as always, I just want to make sure you guys know if you have questions pertaining to what we're doing in SI, that's more than fine to ask. I don't want you guys to feel like you can't ask questions. It's just more like if you had something that required more time, maybe to save it. But maybe I'll try to end SI with, 5 or 10 minutes to spare and hopefully leave a few room for anybody who had any burning questions. Okay, so let me recap of the things that we've done real quick and then kind of catch you up to speed where we are. And obviously, I won't see some of you. You'll be taking your exam before I see you again, right? So we already went over our metric system when we did the packet. We went over this intro to anatomy by me kind of posting an announcement with some of my notes. So we didn't spend a lot of time doing that in SI. We went over the metric lab packet. We've gone over the cell. We did the microscope lab, the mitosis lab, and then we've done the mitosis model, the cell model, mitochondria, phospholipid bilayer. All of this we've done here in SI. And then today, my goal is to do this meiosis lecture. So that's our goal for today, as well as doing these, the tissues packet I've already done, tissues model I've done, and intestine model. So we're just hoping to do these bone, blood cells, skin, and neuron. So basically, the only thing I'm not going to be able to cover with you guys in SI is this histology lecture and the integument lecture. Just to be very clear, both of those will be on your lecture exam number 1. I just haven't had the time with doing all of the lab stuff to also be able to cover both of those lectures. But they're both pretty straightforward on the test in the sense of they're both from a PowerPoint. You have the PowerPoint slides. And they're both like the histology lecture is very much tied to the packet. And if you understand how the tissues look, that will help you a lot with that'll help you a lot with the histology lecture. Okay, so again, histology and integument would be the only things that we would need to go over like by ourselves that haven't gone over in SI. Okay, and then remember there's some example problems for your practice over the weekend. I know some of you have, I believe, your test. I think some of you, the Knudsen students would have your test on Tuesday. So I know that Monday I will see you guys in SI. So if you're a Knudsen student, hopefully we will on Monday's session. So I'll still have Monday 4:45 to 6:45. During Monday's session, let's plan to do some review. So we'll do some review questions together and I'll review some of the lectures. And if you want, again, if you're a Knudsen student, if you want, I can do some integument lecture. Or for those of you who are a Bender student and you maybe have questions on the lab, feel free to come and I'll try to leave some room for that. So we'll leave Monday's session as a review for the people taking the lecture exam on Tuesday, Knudsen students. Study resources module. Remember to check out your study resources module for your practice exam PowerPoint for the lab exam. This is the PowerPoint slides I've gone over with you. Here's some sample questions for the lab exam. Here's a list of what you need to review for the lab exam. And this is that tissues checklist or the study guide that I've shown you guys. I think I've shown all this to you several times. And then lecture exam number 1, practice problems. There are 50 practice problems here. They are helpful. They are, in my opinion, when I took the class, I thought they were a lot easier than the test. And so I remember going into the test and thinking to myself, wow, I'm really glad I didn't like just go off of the practice questions, because I would have probably not been successful on the test. So I don't want you guys to kind of think it's going to be this easy, but at the same time, I don't want to take away 50 practice questions that would potentially help you. So just know that that's not accurate to the difficulty of the test, but at the same time, it's better than nothing to kind of quiz you, okay? On future exams, we have any questions at all? So remember, all of your models will be on the test, right? So let's go through those. So again, if you guys have questions, feel free to interrupt me, raise your hand, or put your questions in the chat. All right, so for this model, this is the bone model. You won't have this picture. So this picture here won't be on the exam. It's just a really helpful picture because it kind of shows you the different osteons and how each of those osteons would look in the bone. But what we test you on is this model here, the one on the blackboard. So it's just one osteon, and this is how we're going to test you. And again, it'll be the same exact model on the test. So we're not going to be using a different model than this one. It'll look just like this. So what you see over here looks kind of like a cell. It's like blue and orange. That cell is your osteocyte, right? So that's the cell in the bone. And then it sits inside of these houses, these kind of empty like circles, right? And those are your lacuna. And so how do you identify the difference on the lab exam? If it's empty, it's the lacuna. And if we're pointing to it and it's full and it's the orange and blue, that we want you to identify the cell. Is that clear to everybody? So if we are asking you for the cell, we want this orange and blue. If we're asking you for the lacuna, it's going to be empty without the cell in it. These here, the cracks, are the canaliculi. Okay, these are the cracks. They basically supply the cells with nutrients. And then right here in the center, these are our blood vessels and nerve, right? And so that's the Haversian canal. It can also be called the central canal. This is the blood vessels and nerve. If something has 2 names, I know I've said this already, but if something has 2 names, you guys do need to know both names. And the reason you need to know both names is because on the test, if it was a multiple choice, right, maybe only one of those names is part of the multiple choice, and it's not the main name that we use like Haversian canal, but it's instead called central canal. So make sure you guys go over both of the names.
I have a question.
Yeah.
Before you like switch slides or erase them, can you tell us so we can make sure that we have everything?
Sure, that's a great idea. I will do that. Yeah. And then there are these like rings. Hopefully everybody's seeing that. These rings that go around. Each of those would be a lamellae. And usually we call them rings. So I'm trying to put in parentheses, maybe how we'd word it on a question. What are the empty spaces? What are the orange and blue? What are the rings? What are the cracks? Does that make sense? What we should also be pointing to it, but sometimes the question will give you guys more detail because basically that way everybody's clear on what we're pointing to. Does that make sense? If for some reason someone didn't really understand what we were pointing to on the lab exam with our little piece of tape. We use a piece of tape. So the entire structure is an osteon or a Haversian system. Okay, and that is everything for this model. And so you can go ahead and take a picture. All right. I don't know how long to wait to take a picture. That's probably good, right? Everybody got it?
Yes.
Okay. All right, so that was bone. So that's everything you needed to know on bone, okay? And then I'm just gonna look at our checklist just in case I forgot something, and then we'll do our blood cell models. So this is your blood clot. For blood clot, the fibrin is already labeled for you. Fibrin are these like hair-like strands, and then we have our thrombocytes or platelets. And those are the only two things that you need to know. So thrombocytes are also called platelets. Fibrin are these little strands, and then the entire structure would be a thrombosis, which is also called a blood clot. Okay, you can take a picture of that. Okay. All right. This is the red blood cell or the
bro, please, I was unable to take the picture, please.
Okay.
Did you label this one? Okay. Is that all?
Yeah.
Okay. Thank you.
Yeah. Okay, and then this is our red blood cell. Not a whole lot on this one, just know that it is the red blood cell, also called the erythrocyte. Its function is to transport oxygen. And then its shape is biconcave, the biconcave disc. So those would be the things that you would need to know about this model. Just that basically this is the red blood cell and then the function of it and the shape. So that's everything on that one. All right, I'm going to move on to the next model now. Okay, so these are the white blood cells. So you have two different types of models. You have these ones that are very small and kind of weird looking. I mean, in my opinion, they're weird looking. And then you have these ones that are a lot prettier. We will use both of them on the exam. So we use both types of models, these ones and these little ones. Okay, so just be prepared. Both types of models could be on the exam. So I'll make sure to label each one. So this is your lymphocyte, okay? This one is your neutrophil, and this is the basophil. The neutrophil always kind of has this appearance to it where it has these like three little strands that are connected, right? And lymphocyte is always kind of like a little, like a mostly a blob, basically. And then basophil has a little bit of a variety. As you can see in this, it kind of looks like a stomach. So then we'll look at the other example of the basophil. So basophil is kind of weird because it has this appearance. Oops, I don't know what's going on with that. It looks, just do a little, it looks like this, right? And then we'll look at the other example of basophil. All right, so everybody get that? Lymphocyte, neutrophil, and basophil. Okay, here we have the monocyte and then the eosinophil. Eosinophil is, in my opinion, like the easiest one. It looks like the letter E. Can everybody kind of see how it looks like an E? Because it has this like little line here. And it kind of looks like an E if you left out the middle part. Yeah. So that's for eosinophil. And then monocyte kind of looks like a bean. Yes, monocyte. All right, so that's it for this one. I'm going to go label the little ones now. All right, so for these little guys, some of the stuff looks very similar, which is good, and then other stuff does look different. So this is going to be your L for lymphocyte. This is your N for neutrophil. And then this is your monocyte here. So the monocyte, in my opinion, looks like a bean. It has a bean shape. That's how you can remember monocyte. Like I said, neutrophil always has these three little lines. Lymphocyte is always kind of round. Basophil here, like I said, it's the one that looks different. You guys see how it looks pretty different than this one? So it kind of has these two appearances. One time it looks like this, and the other time it kind of looks like this stomach thing. And then the eosinophil always looks like an E. Does everybody see how it's just sideways now, but it's still that E shape with the little tiny narrow part here, right? So that's everything for this model. So now I'm just going to write on the side the other things you need to know for the test. So in addition to identifying them like we just did, where basically I labeled which one was which, you need to know most common to least common, right? And remember we said that the way that you know that is never let monkeys eat bananas. So never would be neutrophil. And this is the most common because it's the first part of the mnemonic. Let would be lymphocyte. This is the second most common. Monkeys is monocyte. And this would be like the third most common. And then this is eosinophil. This would be the second least common. And then basophil would be the least common. You also know you need to know which ones are granular. So granular is everything ending in phil. So that would be neutrophil, basophil, eosinophil. Agranular are the site. So that's monocyte, lymphocyte. Then you also need to know function of the white blood cells. So for never let monkeys eat banana, we have a matching mnemonic, BAMF. BAMF, it's not technically a word, it's kind of easy to remember BAMF, it looks like. Does that make sense? It's just something you kind of remember. If you can't remember that, it's just they go next to each other. So it's so that you remember that neutrophil phagocytize B for bacteria, lymphocyte is A for antigen antibody complex, monocyte becomes M macrophage, right? Because M and M, eosinophil phagocytizes parasites, P for parasites, and then basophil secretes histamine, H, and heparin, H. Okay, so those are all of your different functions of the white blood cells that you need to know for the test. So again, on the lab exam or the lecture exam, but mostly lab exams where we ask these, you need to know commonality, granular, agranular, and functions, as well as identifying them, right? Identification on both sets of models. And I already showed you the other models. All right, so that's everything for white blood cells if you guys wanted to take a picture. All right, we good? Everyone got it? Okay. I'm going to move on to our next model, okay? I believe it's neuron. Okay, so it's everything for blood cells. So make sure you guys spend some time on that. I will say every lab exam, we have those on there. So They will definitely be tested. Okay, so for the neuron, what do you need to know on the neuron? So again, this entire model would be the neuron, and then what are the different parts? So the centermost part of the neuron, right? This kind of whole area would represent the cell body, right? I love this pink. Let's see if I can get the color here. This would be your, I'll just write N, okay, N-U-C, nucleus, right? Everybody kind of knows nucleus now. This is the axon hillock. It's kind of this like triangle portion here is the axon hillock. This is the axon. And then coming down over here, that kind of meaty looking orange thing is also the axon. The covering around the axon, this like yellowish color, is the myelin sheath. So that covers or wraps around the axon. And let's cover those next topic. And then these little white structures, kind of like little white, I don't know, hairs, those are dendrites. So you might be wondering, what are all the little orange structures? So those are coming from another neuron. So they are telodendria. Telodendria from another neuron. Remember, telodendria and synaptic knobs, those are the end of a neuron. Telodendria. These are coming from another neuron. So I say that because usually on the test, students will mislabel these and say that they are dendrites. So just to be clear, the orange structures are not dendrites. The orange structures are telodendria. Good. The little round knob here at the end of the orange structures, those little round knobs, that is the synaptic knob or axon terminal. Axon terminal. Okay, so just to be clear, students get confused. They mislabel telodendria as dendrite. Dendrite is the white, telodendria is the orange. Okay, and that's everything for this model if you guys wanted to take a picture. Somebody asked,
Oh, sorry.
oh no, you're good. Somebody asked in the chat, will the other white blood cells be nearby? No, we put all the model, we put all the white blood cells, not white blood cells, we put all the models away when we test. So no, we're not going to have them side by side so you can compare. You guys just need to practice looking at the pictures and differentiating. Yeah, sorry, you had a question.
How, like, a lot of this stuff was never mentioned like in the lecture, so how are we supposed to know all of this if we don't, like, if we can't make it to SI one day?
There should be, that's why we have a list. So like for this one, these lists are your key of everything you're supposed to be responsible for knowing. So like on here, it explains all of basically what I said, but it's on like a document within the folder.
Okay.
So
Yeah.
it should, the resources for what you need to know aren't only in SI, like you should be able to for sure know what you are supposed to know in other places. The first unit is a little bit different than the other units. The other units are, the models have numbers on the rest of the like semester. We have numbers on the models. It's just that these models don't have numbers. So like the first models we use, and it's just coincidence. It's not like anything malicious. It's just that all the ones we use for the first unit don't have numbers versus the other ones later on, you'll have like a number like 39 in XYZ, and then you have a list of what you need to know. But we still try to give you guys the list to make it clear on each link of what you would need to know on those models. And then the links are all found in the module.
Okay, thank you.
Yeah. Okay. I don't have the X on my screen. It might be an X on your screen. Somebody said, click the link, click the X. I think I clicked it a while ago. I didn't know it was showing for you guys. I was trying to get it to go away. Okay. I am going to erase this one and we're going to do a new model, okay? Will we label telodendria or the knob? So like if on the test it's asking you for telodendria, you're looking at the little, like the straight part. Is that making sense to everybody? The straight part, the little circle at the bottom would be the knob. Does that make sense? So if we have a little piece of tape hanging out at the end, that would be the telodendria. And if it's pointing at the very base, the base is the knob. Okay, here is another neuron. So this is like the same stuff that you would be labeling. We might use this neuron. So this here, each of these little guys, like we said, what are those called? They're the telodendria. What telodendria? What are the round parts? The round parts are the synaptic knob, axon terminal. This down here would be the axon. And then the axon terminal is this part here, that middle portion where they kind of meet. Terminal. And you don't really need to know functions. We don't really ask you functions on this model. These would be dendrites. And then nucleolus is at the very center. And you don't need to know any of this surrounding stuff for this model, for this test. And on the next test, we have you guys know more of the stuff in the middle. And then the whole entire middle part, right, would be our cell body. Yes, axon hillock. Sorry, everybody was saying axon hillock. Very good. Good job, guys. Good. So that should be everything. Good. Everybody got that? And then we have one more neuron, and then we'll do
Oh, it's because I'm like a part of like this student like instructional thing.
Mona,
I'm like trying to get it.
I think your mic might be on. I'm so sorry.
Oh, I'm so sorry.
No, you're good. You're good. All right. And then the last one here. Yes, if somebody asked about the different names, synaptic knob versus axon terminal, you guys need to know both names because if it's multiple choice, you would need to identify either of those names, right? So axon terminal or synaptic knob. If it is a fill in the blank, usually it's sufficient to write just one of the names, but for multiple choice, you do need to know both, right? Because we could use either. This neuron looks a little bit different, but again, this is one that we use on the test. So that's why I'm just labeling it one more time so you guys have it again. This would be the axon hillock. Thank you guys for noticing that. Axon hillock. And then in the center here is the axon. And then the branches that come off of the axon are the terminal branches or axon collaterals. So they're basically when the axon branches, we call it terminal branches or axon collaterals. The little like silver, let me see if I get a different color. These little silver like feet. Does everybody see those little silver feet? Those are our telodendria on this model. So remember how we said telodendria are the end of the neuron. And then at the very end here, we have these little knobs again, right? The little circles. And those are our synaptic knob. or axon terminal. Good. So I'm all done labeling that one if you wanted to take a picture.
I have a question.
Yeah.
So for the exam, are we going to have all the different neuron modules for the lab exam, or is it just one of these many neurons, if that makes sense?
It'll be one of these that I showed you guys. So like, we're not gonna bring out a different one, but if we have multiple ones in like the Google Drive, or it'll usually be in the lab on the tables, but that'll be one of these three. Does that answer
Okay.
your question? So not like a different one, but one of these three. That's why I'm showing you on all three. So you guys can see across all three how they look, but you need to know all three.
Okay, thank you.
Andrew, you shouldn't be tested on nodes of Ranvier for this test. That should be your Unit 3 exam, but if you have a list somewhere that's asking you to identify that, I'll just go ahead and label it for you. You shouldn't have to identify that this time around, but I'll label it for you. So the indentations in the myelin sheath are nodes of Ranvier. So again, this was myelin sheath, like the yellowish. And then those indentations are nodes of Ranvier. Those little like indents. Good? All right, so that's everything for the neuron. Any other questions? Just go ahead and do our skin model, and then we'll do the meiosis lecture. So for the skin model, like you guys can see, we have two different models. So there's this model of the skin, and then there's this model of the skin. So for this model of the skin, we really only test you on a few things on this. Then the other model is most of the other things. So the main thing that is special about this model are the two types of sweat glands. So remember how you guys learned about sudoriferous sweat glands, and hopefully you've already taken a look at the lecture, especially if your exam is on Monday or Tuesday. Sudoriferous sweat glands are the water sweat glands, right? And then you also have your sebaceous sweat glands, which are the oil sweat glands, right? There's a little bit of lecture, but this is something that you need to identify on the model. So the sebaceous sweat glands are for oil, sudoriferous sweat glands are for water. So the sebaceous is going to be this purple, like bulby thing that's hanging off of the hair follicle. So that's your sebaceous gland for oil. So this would be the inside of one, right? That's the inside of one. Now you see these two white. Everybody see the difference between these two white ones. This white gland, I need a different color. Okay, yellow. This white sweat gland goes straight to the surface of the skin. Does everybody see that? This white sweat gland attaches to the hair follicle up here. Yes? So there are actually two types of sudoriferous sweat glands. The first type of sudoriferous is called apocrine. Apocrine sudoriferous is a sweat gland that attaches to the hair follicle. And so we're going to label this one number 1. And then 2 is merocrine, and merocrine sudoriferous sweat gland goes straight to the surface. The way I remember it is apocrine releases smell. It's smelly. So if you think about like the areas that have body odor, like your armpits, right, groin, those have apocrine glands and they have more smell. Merocrine is going to be areas where you sweat, like your arms or your palms of your hands, that don't necessarily have hair follicles, and so they don't get as like strong of a smell. Does that make sense? So that's how I remember it. Basically, the stronger smell is associated with the apocrine hair. So hair, 1 apocrine, and then merocrine over here. 1 is apocrine, and then 2 merocrine. So that's the main thing we want you guys to identify on this model are the different types of sweat glands, apocrine and merocrine sweat glands. We might ask you for the Pacinian corpuscle, because you can easily identify that on this model, Pacinian corpuscle. Good. Pacinian corpuscle. And then these would be Meissner's corpuscle. But again, the main thing we're testing you on this model are the different types of sweat glands. What's the function of the Meissner's corpuscle? Light touch, right? What's the function of the Pacinian corpuscle? Detects pressure. All right, and I'm done with this model if you guys wanted to take a picture, and then we'll go do the other model. This, by the way, I guess I'll label one more thing. This is your hair follicle, and this would be hair. All right, I guess now I'm done again. Good. Everybody got that? Okay. All right. So let's do this big model. So the first thing that we can look at, and I'll try to zoom, but not that close. So you guys can see the 5 layers of the skin, right? So on this model, we label the 5 layers of the skin. So it starts out with this bottom layer, right? This dark blue line. That's what makes the skin pigmented or non-pigmented. And remember our mnemonic, right? B, boys, say girls like chocolate, right? So this was basala, stratum basala is that bottom layer where it's that dark blue line. So I can label it also from the top right here. Do you see how it's the dark blue, like that lines kind of this area? So that's stratum basala. And I'll show you a different picture that shows it in another angle too. Okay, what was the next layer called? Boys say, so it was spinosum, right? So everything between the dark blue line and that other pale blue would be spinosum. So this is all spinosum, spinosum. This little layer here, everybody see this kind of like pale blue? That's going to be granulosum, G. So my font is really big when I write it, but granulosum, right, is that pale blue line. And then, so S, G, and then what's next? Lucidum, right? So the white, the 2 layers of white is lucidum. Very top layer is corneum. Okay, so boys say girls like chocolate, basala, spinosum, granulosum, lucidum, and corneum. Good. The little bumps, see all these little bumps over here look kind of like lumps or towers. Those are dermal papillae. They're lumps. Good. You guys see this muscle here? There's a muscle that kind of goes alongside the hair follicle. That is called your erector. Erector pili muscle. So it does have a function. So the function of the erector pili muscle, I can figure out where to write this. Can't really see that very well. How about here? Function of erector pili is to make the hair follicle stand up straight and goosebumps. So it gives us goosebumps. So if I write out a function, I'm trying to give you guys the most commonly asked functions on the test. Good. So function of erector pili muscle is to make the hair follicle stand up straight and to give us goosebumps.
Brooke, do you mind using white for all the labeling? I don't know if anyone else having the issue, but it's just easier to see the white than the red.
Yeah, I'll try to use white. Sometimes I did white and then I couldn't see it, but
Oh.
I'll keep working with it. I had one model and then I was starting out with white and you couldn't see anything, but I'll try to go for white first and then go from there.
Thank you.
Yeah. So the Pacinian corpuscle detects pressure. I've said that like 10 times now. Hopefully you guys are getting it, but if you guys get the Pacinian corpuscle, we'll definitely be asking you like the function, right? Meissner's corpuscle are always going to be towards the top surface, right? Meissner's corpuscle, all of these guys are all Meissner's. See, that's the problem with the white. You see how you like lose the white if I have like another color as the background? So then like I have to change to a different color, but I'll try to work with it. So that's Meissner's corpuscle. Remember the function of that one is to detect light touch, right? To detect light touch. So hopefully I have enough room to do that. Light touch. Hair follicle is surrounded by the sebaceous gland, right? So this is sebaceous gland. The hair follicle are these little guys that stick up here at the top. You don't need to identify the sweat glands on this model. We will ask you on the other model to identify the sweat glands. So the merocrine and apocrine, which I already showed you merocrine and apocrine on the other gland. But sebaceous gland, we could ask you on this model. Does that make sense? Sebaceous gland is very easy. It's always around the hair follicle. But the apocrine sweat gland and the merocrine sweat gland will ask you on the other model if we ask that. Down at the bottom is going to be your hypodermis or subcutaneous. Remember that that is the layer that has the fat in it, right? Equals fat layer. Good. It's all of this bottom layer, all of these little globules of like yellow is the hypodermis. I believe that that's everything on this model. I'll show you the layers of the skin again on another model. I can try to relabel these. So this is B is that bottom layer. S is this light, G, L, and C for corneum. Good. That should be everything for this model. Oh, I accidentally zoomed out. Whoops. Ah. Oh no, I don't know if I can get it to zoom at the right spot again. Did everyone get it? I think my labels are pretty close there. The Meissner's are always these red circles at the top. Somebody asked, does that change? No, the Meissner's are always the red circles at the top. Good. All right. Let me look, we'll do the layers on this other view, and then we'll switch over to meiosis. Remember, meiosis topic for your lecture exam is what I would say is the hardest probably topic on the exam. So I'll do my best to kind of cover that and hopefully simplify it. We'll do a few outlines for that, but I'll just quickly label this. So this is the dermal papillae. The next layer, that dark blue, would be your stratum basala. the dark blue here. The next color, it's kind of hard to see here, but there's like two new blues. There's this blue and then there's this blue. Those are both different, right? This one here and then this one here. So it's stratum spinosum and then stratum granulosum. And then we have the two layers of white, stratum lucidum. And then lastly, our pink layers over here, stratum corneum. Good. Yes, this is just a different angle of the same one. Leonella, you said you had a question?
Yes. I see that you labeled stratum lucidum, but I thought that it was only present in thick skin, like the palms of hands and the soles of feet, but there's hair follicles there, so I'm confused.
That is a great point. I never thought of that. It's just because we want you guys to learn all the different layers. If you look, part of this model doesn't have like hair follicles, so maybe they were thinking like, I think they're just trying to get students to understand all the different things that could be found in the skin, even though maybe this isn't perfectly accurate, like you said, where there's hair follicles. But good critical thinking there. But it's just because we want you to be able to label all the different parts. So I think they were trying to incorporate everything in one.
Okay, thank you.
Yeah. All right, we good? Any other further questions on lab before we move into kind of the lecture side of things and working on the meiosis lecture?
Will the math, like the metric part, be part of the lab test or the exam test? The lecture
Sorry,
test?
what was your question? The first part of it?
The metric part,
Would
would that be part of the lab or the lecture exam?
That'll be on both of them.
On both? Okay.
Yeah, so for the lab part, we might have you measure a line or weigh something or like measure a liquid and then convert it. So like if you got 5 centimeters, what is that in nanometers? Does that make sense?
Okay. Yeah.
For the lecture, it'll be like written out. So 5 nanometers, like it'll be completely written out for you. So what is 5 nanometers in micrometers? Does that make sense? So then you just have to know your abbreviations and then set it up for the.
Okay.
It'll definitely be on both though, because I know on my lecture test, I had multiple ones for metric. And then also on the lab test, I had multiple. So you can expect to see it in places.
Okay. Thank you.
Yeah. All right. Any other questions before we move on from lab?
Not to drag you back. Is there any way you can do the mitochondria again for the model?
The mitochondria model again?
Yes.
I believe it's just a few things. I should be able to do it really quickly. I think I had done it in SI a little while ago, but I'll try to go through it really fast. See, my internet is a little slow. So basically the whole thing is your mitochondria. Each of these kind of folded areas, the areas that are kind of folded up would be your matrix. Matrix are the folds. And then the crista are all of these ball-like structures. Good?
Is that all? Okay, let me take a picture. Thank you.
Yeah.
Thank you.
Yeah, no problem. Okay. All right, so for meiosis, we have the overall like outline that we had talked about with mitosis, right? So hopefully at this point, mitosis is pretty easy for you. So mitosis compared to meiosis, we started out with 46 DNA strands. And remember, they would have been chromatin in mitosis. We go through S phase and they become 46 sister chromatids, right? Remember, they each have a centromere, and then they each have a kinetochore, right? Joined together by a centromere. So that was a sister chromatid that we make in S phase. And then we're going to go through the phases of prophase, metaphase, anaphase, and telophase, which we call PMAT. And then we end up with 46 and 46. Each of those, once again, would be DNA chromatin, right? So that's kind of the really quick recap of mitosis. When we go through meiosis, it looks similar in the beginning, but then it gets different as we go through it. So in the beginning, we have 46 DNA chromatin, just like in mitosis, because really mitosis ends in one of these cells, and then that one of those cells ends up going over and becoming a sex cell. So this happens like basically when a baby is forming, some of the mitotic cells specialize and then go over into meiosis, if that makes sense. So we have the DNA chromatin, and it goes through its S phase and it forms tetrads. So these are called tetrads, and that's 92. Just like over here when we went through S phase and we formed sister chromatids, now we're going to form tetrads. Tetrads come in a group of 4. So tetrad, if you speak like Spanish, it sounds kind of like 4, right? So this comes in a group of 4, and they're still joined at a centromere, and then they each have a kinetochore. So tetrads, there's going to be 23 tetrads, because 23 times 4 equals 92. We're going to go through 2 phases. There's going to be prophase, metaphase, anaphase, telophase. Number 1, which divides that 92 into 46 and 46, and we call these dyads. So the end result is dyads. We have 23 dyads and 23 dyads. 23 times 2 equals 46. 23 times 2 equals 46. Now those are going to go through a prophase, metaphase, anaphase, telophase number 2. And number 2 brings us to 23, 23, 23, and 23 individuals. And these are DNA chromatin at the end. So it's kind of like mitosis in the beginning, but then it goes through it a second time. And the big key difference is that when we go through S phase, we form groups of 4 that split once into a group of 2, right? And then they're all by themselves. Whereas over here with mitosis, we basically have that sister chromatid, which is just a group of 2, and then it splits into a group of 1. Does that make sense? So we take an extra step because we make tetrads. So we're going to go through what happens in each of them, and then the different terms you would need to know for testing. This first starting cell is the diploid cell. So a diploid cell, we can abbreviate that 2N because it has 23 basically times 2, and N equals 23. Each of these would be a haploid sex cell, N, which equals 23, right? So we have the haploid and the diploid. Diploid would be the starting cell, and haploid is the ending cell. All right, so now we're going to go through what happens in each of the PMAT stages, just in general for meiosis, and then I'll talk specifically about female and male. So we'll do our best to kind of cover everything in an hour. We'll see how I can do that, but it'll be basically review. In a sense, I just encourage you to keep your notes out and just keep track and make sure you have everything that I'm getting down, and then add in if I say anything new that you don't have. If you have a question or something has been confusing you, you're welcome to raise your hand, ask, or interrupt me, and then we can try to address that, okay? So right now what we're gonna do is we're gonna talk about PMAT number 1, okay? We talked about the S phase, and then now we're talking about PMAT number 1. So for prophase number 1, we're going to have basically the cell, and then we're going to have these tetrads all over the cell, kind of at random, right? Each of them with a kinetochore and a centromere. And then for metaphase number 1, they're going to line up at the equator. So we're going to have those tetrads lining up at the equator. So the movement of the tetrads to the equator is called synapsis, okay? Movement of the tetrads to the equator. So basically, prophase 1 to metaphase 1, this movement itself is called synapsis. Okay, so that's a term you need to know for your test, okay? During synapsis, DNA or genetic exchange happens between the tetrads, and we call that chiasma. Happens between the tetrads. So they're kind of like neighbors with each other, all these little, the tetrad neighbors, and we call that DNA exchange that they're doing chiasma or crossing over, right? So on your test, you might be asked, what is the movement called? And you would say synapsis. What is the actual genetic exchange between each of those tetrad pairs or tetrad quadruplets, I guess? And you would say chiasma or crossing. Does that make sense? So we have the tetrad moving from kind of at random to the middle of the cell. So then we go to anaphase number 1. For anaphase number 1, we remember that the everything pulls apart. So in this case, the tetrad is now pulled apart. It used to be a group of 4, but now the centromere breaks, just like it did in anaphase of mitosis, and we have dyads. So there are dyads at opposite ends of the cell in anaphase 1. When the dyads form and the centromere breaks, we have another word that you need to know, another term. We get recombination. And recombination is the term for the new genetic code to be passed on. When you go from anaphase 1 to telophase 1 would be the cytokinesis or the division of the cytoplasm. We have dyads on one side and dyads on the other. Okay, so this is 46 as 23 dyads, and this is 46 as 23 dyads. So that's everything for PMAT number 1. But instead of going through another S phase, these both will go straight into PMAT number 2. Okay, so for prophase number 2, we're going to have those dyads basically all over the cell at random. And remember, we have this happening twice, right? Because we have 2 products from the previous telophase.
It's okay. Can you just stop there? I just want to take a picture of that.
Yeah, no problem.
Thanks.
Did you get the first one?
Yeah, I did. Thank you.
So each of these is now going to go through its own PMAT2. So we have metaphase number 2. So these are going to line up at the center, right? Line up at the equator. And I'm just drawing it twice to kind of keep you remembering this is happening twice over. Then we have anaphase 2. They're going to pull apart, right? The dyads are now pulling apart to opposite ends. And I'm just keeping the drawings really simple because for mitosis, I already went through all of the different structures. Just know you have all of that same stuff again. The centrioles, centrosome, the asters, the spindle fibers, right? All of that stuff is still here. I'm just not going to spend our time redoing all of that. And then for telophase, right, we're going to end up with 23 and 23 each of these, right? And they're 23 singles. And what do we call these? Haploid cells. Okay, and that's the end result of meiosis 2. Someone wanted me to go back here, I think. Good. Got it. And then this would be our last phase. So again, we start out kind of at random with tetrads. We move to the equator, which is called synapsis, at which time the genetic information is being exchanged. Then we pull those apart into dyads, and that's called recombination. And then those move on into prophase, metaphase, anaphase, telophase number 2, or meiosis 2, when we actually get prophase 2, metaphase 2, and then they pull apart right here individually and then become 4 haploid cells in telophase 2. Good? So now let's talk about male and female meiosis and why they're different, and then the terms you would need to know for testing. So for male meiosis is called spermatogenesis. Okay, it basically, there's a few things that we look at. First, we say that the process of male meiosis is going to look identical to what we just talked about. So everything we just said will be the same for males. So the whole process I just outlined, the simplicity of my meiosis, each of these will be a sperm haploid cell. So what is the end result of male meiosis? The end result of male meiosis is 4 haploid sperm. Does that make sense? 4 sperm, each with 23 in it. And then we talk about a little bit of the anatomy of males so that you guys can answer some questions kind of about how sperm is stored in the testes. So this is the testes. Inside each testy, we have little empty houses, and we call those empty houses lobules. Okay, so these would be testes. Each of the empty houses is called a lobule. Okay, so these are the empty houses. Inside of every lobule are seminiferous tubules. So these are kind of inside seminiferous tubules. These are made out of spermatogonia. And spermatogonia are specialized cells. So if we kind of draw a little seminiferous tubule, it looks something like this. And then lining the seminiferous tubule are our little spermatogonia cells. So these are going through meiosis. And at the end of the result, we have these little sperm cells that are formed, right? These are our haploid sperm that are formed. These spermatogonia cells that line the seminiferous tubule, all of these are little cells here. These cells are spermatogonia. They are the only cells in the body capable of mitosis and meiosis. Okay, and so these are actively going through mitosis and then meiosis. Remember that males are fertile until they, basically almost until they die. So That's because their sperm cells are constantly generating more sperm cells, and then those are going through mitosis. Kind of like how I showed you, there's mitosis, right, which forms the 46 and the 46. Yes, this is mitosis. With male spermatogonia, they're going to go through mitosis, and then they're going to send one of these to go through meiosis, and then this one will then go through mitosis again, right? And then send one of these to go through meiosis. Does that make sense? So it's like one keeps replicating forever, and then one goes through meiosis. So that's spermatogonia. There's a storage point inside of the testes for the seminiferous tubule. So they all kind of join together inside of the testes. And that's called the ret testes. So the ret testes, this is the storage for seminiferous tubules inside of the testes. This is where they collect inside the testes. Then they actually leave the testes and they come out before leaving into what are called the epididymis. So the epididymis. Let's see, can't read that. Okay, that's pretty good. So this is the storage for sperm outside the testes. Good. So we have two epididymis, two ret testes. Ret testes would be inside the testes, epididymis outside the testes. Seminiferous tubules are inside of the lobule, and they're made of spermatogonia. Lobule is the empty house inside of the testes. Good. So that's everything for males. Does that make sense? Males go through meiosis the same way as what we described in the beginning. They have these specialized kind of cells, the spermatogonia. You need to know everything on the screen for the test, right? You need to know what ret testes is. You need to understand what the epididymis is, what seminiferous tubules are, what the lobule and spermatogonia are. Good? Any questions at all there? Okay, so I want to cover females. So when we talk about female meiosis, it does get a bit more complicated. So I'm going to go a little bit slower, but I'm not going to go super slow, but I'm going to try to make the point make sense. So if you're thinking about a female anatomy, you have basically the uterus, and then you have the little ovaries over here, right? Female uterus with the ovaries. Okay, the ovaries inside of one ovary, so we're going to look at one ovary. Inside of the ovary, we have eggs. So there are a lot of them, right? There's actually like, I believe, thousands, but I'm just going to kind of fill it up so we can picture it. And each of these eggs lives in a house. So the house is the ovarian follicle. So this contains the egg, and then the egg is the ova oocyte, okay? And these ova or oocyte are formed while the female is in utero. So basically the second that female baby is born, all of these ova or oocyte are filling up the ovaries. Each ova, when a female is born, is in diaphase, and diaphase basically is the resting phase. So you could think of it as all of them are sleeping. All of those eggs are sleeping in diaphase, and all of those eggs are all in prophase 1, they're all tetrads. Remember, we talked about 46 duplicate in S phase, right? And they become tetrads when we're talking about meiosis. They become 23 tetrads, right? 23 times 4 is 92. So 23 tetrads. And then we go through prophase, metaphase, anaphase, telophase 1. But they haven't finished prophase, metaphase, anaphase, telophase 1. They're just sitting there in prophase 1, and everybody's just resting, right, in prophase 1. Does that make sense? So every egg, when a female is born, is in diaphase, resting or asleep, in prophase 1 as tetrads. Good. So now we move into the first menstrual cycle. So the first time a female ovulates, right? The first menstrual cycle or ovulation. And so what we're going to see is one of those ova is going to leave diaphase and wake up. So we have wake up and one is chosen. Good. So that's the next page. We're going to go there now. So when one ova is
Can you go back really quick? I'm sorry.
no problem.
Thank you.
Yeah.
Okay.
So when one ova is chosen, so this would be the first menstrual cycle, like a girl getting her period, right? This egg, and then every time that you go through a menstrual cycle for the rest of a woman's life. So when one ova is chosen, so we're going to say each month, right? For each menstrual cycle, first menstrual cycle and subsequent menstrual cycles, this egg will progress, right? It's going to change from prophase 1, and it's going to go all the way to metaphase 2. So this is why it's kind of helpful to draw it out. So I'll draw it out real quick because that's how I learned this content when I was taking the class. So this is now renamed. So when chosen, renamed the primary oocyte. Just think about it like primary, like the most important one because it's the one going through the cycle, and it's living in the primary follicle, in the primary follicle. Primary oocyte living in the primary follicle. So when you think about it, we'll just draw a quick little recap, right? Because this helps us visualize what happens, and there's a really important thing you guys need to know that occurs here. So we have prophase 1, metaphase 1, they kind of line up here at the equator, right? And then we get anaphase 1. They're going to separate into the dyads. And then we still have all the same stuff, just so we're clear. We still have synapsis. Remember, that was the movement. We still have chiasma, which was the genetic, or we'll just write DNA exchange. And then when we get to the dyad formation, we still have recombination. I'm writing M1. If you mean over here, this is metaphase 2. This is M1. So then we have telophase 1, right? And telophase 1 is when we get those dyads kind of become 2 different cells, yes? But the thing that happens in females is that there is an unequal division of the cytoplasm in telophase 1, which causes the death of one of the basically byproducts, which would have 46 DNA. 23 dyads. So one dies, and this is the first polar body. So on the test, what is the first polar body? The first polar body is formed right here in telophase 1, and it has 46 DNA strands or 23 dyads. So after telophase 1, just this one product goes on and goes to prophase 2, where all of those dyads are kind of everywhere, and then to metaphase 2, where those dyads line up across the equator. Does that make sense? So when we're talking about the primary oocyte progressing from prophase 1 to metaphase 2, what do we have happen? We have the formation of the first polar body. Does that make sense? And hopefully help with me writing it out where you can kind of visualize it. One of them dies off and forms the first polar body. Okay. So we're not done yet, but I'm going to move on to the next screen. If anybody wanted to take a picture or get anything here. All right. So metaphase 2, kind of hold that in your mind. Metaphase 2 is where we left off with one polar body forming. So we're in, just going to go back to this little picture here. We're still in this little ovary, right? In the ovary. So I'm going to draw an ovary again. We had what? The primary oocyte. What stage is the primary oocyte in? It's in metaphase 2. Yes, we just kind of explained all of that. And it's living in the primary follicle. Before ovulation, this is going to grow. So I'm going to change the color. So this becomes the secondary follicle and then the secondary oocyte. So this would be kind of step number 1. And this is step number 2. The secondary follicle and the secondary oocyte, nothing changes with the DNA. Nothing changes with the DNA. So it's still in metaphase 2. And it is just growing in size. The same thing keeps happening. So we go to like step number 3. And this is now called the Graafian follicle. So this would be number 3. So the Graafian follicle. This is huge in size. It's growing very large because of estrogen and progesterone. What phase is it in? It's still in metaphase 2. Nothing has happened to the DNA in the Graafian follicle. Then we get the next stage, which is ovulation. Ovulation is when the actual egg bursts and it releases the egg. So essentially in ovulation, we get the release of the egg, from the ovary, and this was one of my test questions, to the abdominal cavity. So on my test, it asked, what is the first place the egg goes after ovulation? And remember our little diagram here, we had that uterus, the tubes, and then we had, this is basically vagina, cervix. We have those little ovaries that are kind of suspended in the air. And so that egg, when it leaves, it's actually going into the abdominal cavity. It's not entering the uterus or anything yet. Does that make sense? So the abdominal cavity is the first place the egg goes after ovulation. What stage is the egg in? It's in metaphase 2. Now the follicle is still left behind, right? So the follicle is now empty. So this is number 5. So number 5, the empty follicle is called corpus luteum. So the follicle no longer has the egg in it, and we call that the corpus luteum. And then finally, number 6, the corpus luteum shrivels up. So the follicle, so it like shrivels up and basically dies. And we call that the corpus albicans. Okay, so you know how like a corpse is a dead person? Just think of corpus luteum and corpus albicans as being the dead form because the egg has left, and now it's just the follicle that's left behind. Okay, so now we're going to talk about what happens to the egg. All right, so you guys are doing great. Just hang in there. We're kind of almost done. We just have to talk about what happens to the egg after ovulation. Number 3 is Graafian follicle. Good. All right, so once we get ovulation, what happens to that egg? So we have our little kind of, I feel like the diagram is very helpful, especially here. We have the fallopian tubes, uterus, and then we have those ovaries. So this is an ovary. This is a fallopian tube. These are called fimbriae. They have cilia that rotate or move to bring the egg inside the fallopian tube. And then this is the uterus. This would be vagina. This is cervix. And then this section here in the middle is called the ampulla. And this is where fertilization normally occurs. So ampulla will be tested, fimbriae will be tested. Okay, the uterus, cervix, vagina is more for like your reference. So after ovulation, the egg goes into the fallopian tube because of fimbriae. So like I said, fimbriae are kind of like sitting there with these cilia, and they create kind of like a current or water that causes that egg to be attracted into the fallopian tube. Good. So that egg migrates into the fallopian tube. So step number 2, if sperm are present in the fallopian tube, right? So sperm can live up to 5 days. So those sperm have been kind of sitting here waiting in that ampulla where fertilization normally occurs. They're kind of waiting, and they're waiting for that egg to come and join them. Does that make sense? So they're waiting in an ampulla. If sperm are present in the fallopian tube, so we'll write ampulla, fertilization will occur. Okay, so when fertilization occurs, it looks like this. So the actual egg itself has a few parts to it that you need to know. So there's this outer portion called the corona radiata. Okay, the corona radiata, which is a protective outer covering of the egg. Then there's the very center of the egg. Remember, this has our DNA in metaphase 2, right? You guys remember it was in metaphase 2. So what does that look like? Dyads at the equator. How many? 23 dyads, 46. In between there, you have the zona pellucida. Zona pellucidum. And this is the like a fluid-filled area between the corona radiata and the nucleus. Remember, the nucleus has the DNA, right? So when the sperm is attempting fertilization, right, we're getting fertilization by the sperm of the egg, we have this substance on the head of the sperm called acrosome, okay? An acrosome is an enzyme that neutralizes the corona radiata. So essentially, the sperm approaches the egg, it releases this acrosome that allows it to get inside the egg, and the sperm, inside of the sperm, it has its own 23, remember? Because all the way back here, what was a sperm? A sperm was a 23 haploid, right? So that, when we look at this picture here, that is what's trying to get in and meet up with this egg nucleus, okay? So hopefully you're thinking, you're kind of like getting to this next step, thinking, wait a second, so this is the egg, has 46, and then this is the sperm, and it has 23. That doesn't look right. That doesn't look like if those join up, that math is going to work out. And we're still in metaphase 2, right? So like, how in metaphase 2 has the female finished meiosis? Well, that question is a great question. So when the acrosome comes, right, to neutralize the corona radiata, the egg finishes meiosis so that 23 joins with the sperm's 23, okay? So what does that look like? So essentially, like I said, basically you get that sperm, it comes in, it neutralizes the corona radiata, and then the egg is going to finish out its meiosis. So it was in metaphase 2 with those 23 dyads, right? And now they're going to pull apart in anaphase 2. Trying to just keep it really small because I feel like it's nice to see it and we don't need it super big. So telophase 2, this is really important. The same thing is going to happen again. Remember how we had a polar body? So in this part, I can erase this. So in this part, telophase 2, when again the acrosome of the sperm came in, the egg finished meiosis, this is going to form the second polar body. So it's going to finish meiosis, making 23, right? And then, and the second polar body. So that's the end result of meiosis 2 in a female would be 123 haploid and a second polar body. What does the second polar body have? 23 haploid, right? What about when we first talked about the first polar body came at the end of telophase 1, and we said that that was a diploid, right? So the first polar body was a diploid. So the end result of meiosis 1 in a female is the first polar body, so a diploid cell plus a diploid polar body. The end result of meiosis 2 is a haploid cell plus a haploid polar body. Does that make sense? So I know there's a lot going on the screen, but essentially I was trying to fit it all so you could see it all in one place. I didn't have to do a lot of different screens. So we get ovulation, the sperm joins up with the egg. You do need to know acrosome, corona radiata, zona pellucida. And you need to understand that when the sperm enters the egg, we get the finishing of meiosis. Does that make sense? So when does the egg go from metaphase 2 to telophase 2 and finish meiosis? That happens when the sperm gets together with the egg. Okay, and we call that a zygote. Okay, so maybe up here. A zygote is the union of the two nuclei, the sperm nuclei and the egg nuclei. So a zygote is the joining together of the sperm nuclei, which has 23, it's a haploid, and the egg nuclei, which is 23 haploid. What's going to happen after we get that zygote? This will lead to mitosis. So essentially that egg and that sperm now can go through the process of mitosis together because they have 46, right? 23 and 23 led to 46, and they can keep going through mitosis. Okay, so really quickly, what would happen if we did not get this sperm? So what causes meiosis to finish for a female? The sperm entering the egg, right? So with no sperm, so no fertilization, the egg is the uterine lining sheds, right? And that's menstruation. And what happens to the egg? The egg remains in metaphase 2 as dyads, 23 dyads at the equator. Remember, 23 dyads would be 46. And it basically leaves the body in menstruation with the first polar body. So you might get a test question that asks you what leaves the body in menstruation. And the correct answer is going to be an egg in metaphase 2 and a polar body. And a polar body with 23 dyads. Does that make sense? If you were asked what leaves the body in menstruation versus when does the female egg finish meiosis? The finishing of meiosis by the female egg happens when the sperm enters the egg. Good, and we call this fertilization. And this would lead to the formation of a zygote. And a zygote is the very first cell that will become a baby, right? A zygote is going to go through mitosis, and it's going to lead to the formation of a baby. Good. So over and over again, millions of times mitosis until we get billions of cells that make up a human being. All right, any of the areas that I can go back to for you? We have a little bit more time, so I'll be able to leave a little bit of time for questions, and I can go back. I can also draw a quick outline of the meiosis if anybody's interested in that. So again, we talked about the process of meiosis. We went through that, and then we explained that in males, right? In males, we get the same exact process of meiosis repeating. And then when we go through females, we start out with the follicle, all diaphase resting. One is chosen to basically wake up. That becomes the primary follicle, which progresses from prophase 1 to metaphase 2. primary follicle, primary oocyte. We form the first polar body during this process of choosing it and it being the primary oocyte. Then it goes through a period of growth where it becomes the secondary follicle, the Graafian follicle. It gets ovulated when it's so big it bursts, and then the empty one is the corpus luteum, and then it shrivels up until it's the corpus albicans. Yes, the dead dyad would be the very first polar body. So that very, very first polar body right here is the dead dyad. Yes, that's one way of putting it. And then we have, after we get ovulation, we're going to work on fertilization, which is where all the action happens, right? The sperm comes in, fertilizes that egg, and that's when the egg finishes out meiosis and forms the second polar body. Together they join and form the zygote, and then that zygote is going to go on and continue performing mitosis thousands of times. If it's not fertilized, then we get menstruation and the egg and the second polar body leave the body at that point. Any other questions here that I can help you guys with? All right, you guys are free to go if you need to go at this point. If you have any other questions, please stay and ask. I'll be here until 11, so about 15 minutes that I can help you guys. And write me over the weekend if you have any questions at all for the test, and I'll do my best to help you guys. Yeah, no, you're good. Dyad haploid difference. So basically, a dyad is when you're thinking about this. I'll just go back to the pictures I drew. So we have the tetrad in the beginning, right? So the tetrad is like a group of 4. So tetrad is a group of 4. Now, when the 4 pull apart into 2, these would be a dyad is this 2. So tetrad is 4, dyad is when they pull apart into 2. And then haploid is when they eventually become just 1. So that's kind of the easiest way to think of it. Haploid is when they're 1. Dyad is when they pull apart right here from being originally a tetrad. So tetrad goes to dyad, goes to haploid. And then you asked about diploid. So that's a great question. So a diploid is the starting cell. So remember how we talked about 46 goes to 92 and then goes to 46, 46, and then those become 23. I don't really have room, but 23, 23. This very starting cell is the diploid. So we never use the word diploid again. The starting cell would be the diploid cell. And that's just, yes, so haploid is half a diploid, yes. Haploid is half a diploid, but this is not a diploid. These would be dyads. Does that make sense? Yes, zygote would be a diploid. When we get the 23 from the sperm and then 23 from the egg, this would form a diploid cell, yes. Basically, as long as it's the starting cell, so it can either start mitosis. So if you're looking at mitosis, this is mitosis, right? Mitosis forms to diploid. So the end result of mitosis is a diploid, and it starts as a diploid. But when it comes to meiosis, the only diploid is the very starting cell, would be the diploid cell. Does that make sense? Not the second. If this is meiosis, remember how meiosis does this and then goes 23, 23, 23, 23. This is not a diploid. This is a diploid. These are called dyads. Does that make sense? So mitosis, everybody who's 46 is a diploid. Meiosis, only 46, diploid is the starting, and then dyad would be the second 46. Great question. Any other questions?
Yeah, sorry. I had a question about the tissues. The Meissner's corpuscle and the Pacinian corpuscle, what tissue are they made out of?
Meissner's and Pacinian. You actually
Yeah.
don't need that. That's a great question, but you don't need to know. You just need to know their function. You don't need to know what tissue they're made out of.
Okay, thank you.
I've never thought of that. Must be some type of connective tissue, but you don't need to know.
Okay, thank you.
Can you explain the sudoriferous gland? I'm kind of confused with that one. Is that just like honestly just like a different meaning for the sweat gland?
Yes. Someone asked haploid is N, diploid is 2N, just because I was on this, and then I'll answer your question, sorry. 2N, N is 23, so if you multiply 23 times 2, you get 46. Does that make sense where the N comes in? If N is 23, we multiply that times 2, and it's 46 for diploid. Can you ask your question, sorry, one more time about the sebaceous gland?
The sudoriferous gland. That
Like
one-
what?
Is that just like another like meaning for like sweat gland, or is there? I'm kind of confused. Like I understand the sebaceous gland, but I'm kind of confused when it comes to the sudoriferous gland.
Sudoriferous would be the same thing as a sweat gland. It's just that there's two types. There's apocrine, which is like smelly sweat, and then
Yeah.
merocrine, which is like watery sweat. But sudoriferous just means sweat gland.
Okay, alrighty, that makes sense.
Good. Any other questions?
Will we be asked to label the sarcoplasm of skeletal muscle? Because I don't know how to label it.
Sarcoplasm skeletal muscle.
Yeah, it's one of the special terms that we have to know. But
I don't, you shouldn't. Sarcoplasm would just be the cytoplasm. Is that in your packet?
it's on the tissues checklist.
Okay, I don't think so. Let me check. Do you recognize it from your packet or no?
No, from the packet, no.
Okay, then you're good on that one. You don't need to know that one.
Okay, thank you.
Yeah. Other questions?
I have a question.
Yeah.
Can you go over the prototypical cell? I have it labeled, but I just want to make sure I have it correct. I was having trouble with that one.
Are you still there? Oh, sorry. Can you
Yeah.
see my screen or did I not share it? Oh, I did share it. There we go.
There you go. Yeah.
Okay, so
Brooke, when you finish with this, can you also do the intestine real quick?
which one?
The intestine?
Sure. So the black circle would be the centrosome. The orange are centrioles. The black circle is the centrosome. Does that make sense? Like the whole black circle. You don't really need to know a function on that one. This would be rough ER. I don't think I need to write out ER, right? You know what ER is. This has ribosomes. So it's going to transport proteins. Smooth ER. Transports lipids. And then oh, the ribosomes themselves, those little white dots, make protein. And then the holes are the nuclear pores. Okay. That should be everything, I think, and then all the functions as well.
Thank you.
Yeah. Did you get a picture okay?
Yeah.
And then you said the intestine.
Yes.
So the tissue is simple columnar epithelium. The blue dots are goblet cells. They secrete mucin, keep the intestine moist. The big blue structure is a lymph node. This is a capillary bed that absorbs carbs, nucleic acids, and proteins. This is the lacteal that absorbs fatty acids. And then the microvilli are the hair-like structures that line the simple columnar. And then those have the function to slow food down for absorption. And then each of the finger-like structures here, each of those large finger-like structures are the villi. Villi, and they increase surface area. And that should be everything on that
Okay, thank you. Let me take a picture.
Amy, I'm actually out of time for today. I'm so sorry. I think that there might be another recording available in the module under SI previous recordings when I went over the mitosis models. I'm so sorry about that. I actually do have to go today because I have an appointment I have to go to, but maybe I can do that for you on Monday.
A quick question, Brooke. The micro villa, is it you didn't label that?
No, there's not a way to label it, but I explained what it is. So it's the
Okay.
hair-like structures that line it. There's no way to label it.
Okay, all right. Thank you. Thank you so much.
Thank you, guys. I'll see you guys on Monday.
Okay, bye.